I got sort of a crush on the sea slug. It's just dresses well, it glows, and it's part of an incredible diverse group called mollusks. We've probably heard about them, but we didn't know how big they were, or at least I didn't until I started listening to Michael Middlebrooks. And I am excited to speak to a person who's really an advocate for the tiny wonders of the world. Sometimes it's good to get away and go in the sea and just look at the wonders, and maybe that helps you get a little more excited about life. So we know about photosynthesis. We learn it as a kid, right? How plants get food from the sun. But we still have to water them and give them, soil. But what Michael teaches us is that there are animals that all they need is the sun, and they use the sun to convert energy. They get energy from the sun, but they also get sugar from that energy. And corals, as an example, do this really well. But Michael's focus is on these mollusks. And they range from like snails to clams to octopi. Octopi. But he's focused on sea slugs. So everyone's question to him probably when they meet him is like, why? Why sea slugs? And I guess all of us have our little magical places we go when our imagination really kicks in. And I'm assuming that that's what happens with Michael. Michael, welcome to Swell. I saw your TED Talk, and you have such a respect for this world and such admiration for it. But the more you go into the focus of one particular little guy like a sea slug, I'm imagining that you can just stay in that rabbit hole for quite a while. How long have you been studying this particular sea slug? And what does it do for your capacity to see hope in the world and see that some of these, maybe I would call them mollusk technologies, that they can photosynthesize and use energy wisely? Does that give you hope for how we might use this as humans on the planet, or is this more just let's just study invertebrates because they're so cool?
Thank you, Deborah. I've been studying sea slugs for about 15 years. I started studying them in graduate school. I met a professor there named Skip Pierce, who was working on these sea slugs. And he is more of, his area is kind of physiology and biochemistry, and was doing some very, very interesting work with these animals. And I was more interested in ecology, so I started asking him some questions about the ecology of these animals, and he didn't really have the answers, so I went and looked to see what I could find in the literature, what we knew about them, and it turns out we knew very, very little. And when I found that out, that this was an area that we know so little about and was so wide open to be explored, that got me really excited, and that's when I started working on sea slugs, and what really got me interested in them was just that there was so much to learn. Just to summarize real briefly, the way these slugs photosynthesize is they actually sort of steal the mechanism to do it. So organisms like plants have inside of their cells these little organelles called chloroplasts. And when the slugs feed on algae, they feed on marine algae, they will take these chloroplasts out of the plant cells and put them inside of their own cells. And they're somehow able to keep those chloroplasts alive, maintain them, and use them for their own photosynthesis. And so in some cases, like Elysia chlorotica, the emerald slug, they can do that for their entire adult life cycle. The species I study, and I've put a picture of this in my response here, is Elysia crispata. They're in the Caribbean and Florida Keys. They can photosynthesize for 3 or 4 months after a meal. So an incredibly long duration for an animal to maintain photosynthesis and gain all of their energy from doing that. And why I've continued studying these slugs is because they're just so interesting, and there's so much to learn. We know so little. For a lot of these small animals in the ocean, the only thing that we know about them, the only scientific paper out there, is a brief morphological description naming the species. And there's a lot of animals out there that haven't even been discovered or named yet. So there's just so much to learn about undiscovered animals, and even the ones that we know about, we don't know some very basic information about them. So for me, it's very exciting just to see what's out there. Your question kind of hinted at a question that I get fairly regularly talking to people, which is, is there a sort of human application for this? Are we able to become photosynthetic ourselves? And I think the answer to that is probably not. There's some sort of physiological barriers that would prevent that from happening. Just briefly, if you think about how something really big like a tree photosynthesizes, they're able to do it because they have these really flat surfaces to catch light, the leaves, right? We would need something very similar. It's a surface area to body ratio issue. So I don't think that's ever very likely, in addition to a whole bunch of additional biochemical complications. As great as it would be to not need a job and just go lay on the beach to get all of your energy. For me, what's really exciting about this is just learning something new about the natural world and discovering what was previously unknown. There's so many little interesting animals and other organisms in the oceans that we just largely go ignored, and there's so much to learn about them. So we don't know what's out there, and I can't say what might come for this in the future. Maybe there are applications that humans could use, but currently my interest is mostly about discovery and seeing what we can learn.
Hey, Michael, great to hear your voice. And I want to say something on the top of this reply to you, which is that it's so important that we celebrate curiosity and the time a person spends going deep, and for no other reason to go deep and to enjoy that experience. And I'm bringing this in because rarely do we ask great painters or great musicians, the application of their work beyond their work. I mean, we revel in the amazing results of these people's work when they go to the place they need to go to enjoy and to discover themselves, and what inspires them, what comes out is incredible art, and we get benefit that. And I think scientists who go to the smallest kind of study and just revel in the wonder, they become people that understand wonder, and they understand the value of a life that's theirs and the life that they're studying. I think it's a very circular experience because your life is so infused with this wonder, and it makes you the person you are, and who knows what that will yield as a discoverer. Because it's not always a straight line, but at some point you might be in a position to discover something that's helpful for someone to behold, even if it's just a talk. And that becomes its own ripple for people to be inspired to go to the place they need to go to. So I never want to challenge the value of work that doesn't have a direct human application. It's really important for me to say that. But I want to follow this up with a question around the cell that you talked about. I forget the name of it. The slug takes in this cell and puts it in their own cell to become useful in their own body. I'm sure there's a name for that, but I just want to understand that a little more. How do you take a cell and put it into your cell consciously, or is this it's not conscious? Their brains don't do this, right? This is a body-to-body thing. So when they want to absorb the photosynthesis cell, the cell that's responsible for photosynthesis, and that they bring into their own bodies, and you said they put it in their own cells. What is that called, and how do they actually do that? And do we as humans do that? Because I know that if I have I think they call them free radicals, when you have something in your body that's open to be attached. Sometimes it's good, sometimes it's bad. Can you get to the cellular part of this and explain a little bit more about that mechanism? Thanks so much.
Okay, well, I will do my best to answer your question here, but first I would like to say that I appreciate what you were saying about the value of research for the sake of learning, and I of course couldn't agree with that more. To get at your question, what we're describing, this phenomenon here that the slugs are doing, has been termed kleptoplasty. So klepto, the root of that word is like kleptomania, right? Like to steal, and then plasty is referring to the plastids or chloroplasts that they're stealing. So that's the term for what the slugs are actually up to. They're stealing chloroplasts. And chloroplasts are not exactly cells. They're typically considered to be organelles, so a component of the cell from the algae that the slugs are eating. And so the slugs will just take these cellular components and put those inside of their own body. And the way this happens is a little bit different than what you might imagine. So the way the slugs feed is they will puncture a hole into the algae that they are eating and slurp out the cellular contents from that through the hole. So they're suctoral feeders. So they're sucking out the chloroplasts and any other cellular components that are in there. And then the digestive tract of these slugs is not really what you would imagine. It's not sort of just a one-way tube like it is in most animals. It's highly branched, so it has all of these different branches coming off of it. A lot of them form sort of dead ends or cul-de-sacs, and in those areas at the edge of those are specialized cells that are able to engulf these chloroplasts. How exactly they're doing that, we don't fully understand, although cells engulfing components is not all that unusual. Really what we don't have the full understanding of is how they're able to, instead of digesting those chloroplasts or whatever else they engulf, the chloroplasts in this case, how they are able to keep them going. We do see that when they do this, they have a membrane that they put around the chloroplasts, so we can see that inside of there. If this were more of a permanent interaction, in the case of the slugs, we call it kleptoplasty because they steal it from the algae directly. But if it was something more permanent, where these could be passed down to the next generation, you might use the term endosymbiosis instead. So where those chloroplasts to be maintained permanently, and ultimately the chloroplasts inside of the plant and algae cells did arise through an event of endosymbiosis. So the ancestors of those chloroplasts were actually free-living photosynthetic bacteria that got engulfed by a cell a very, very long time ago. And so that's sort of how chloroplasts originally arose, but now they're not considered to be separate cells because they're highly integrated into the system of the plants and algae. We have a similar sort of thing in our own body in the mitochondria. So the mitochondria that are inside of our own cells were one-time free-living bacteria, and now they've become organelles inside of our body. I'm hoping that gets sort of at the answer that you were talking about. Back to the slugs. Once they've engulfed those cells, they are able to keep them. Once they've engulfed the chloroplasts, not cells. Once they've done that, and have them stored in their own cells, they're able to keep them alive for varying amounts of time, and that's really where a lot of the mystery here is. This requires some complex biochemistry that we don't quite understand, but if you remove a chloroplast from a plant or an algae cell on its own, it won't survive very long. And it certainly won't survive for the many months that some of these slugs are able to do it. So that's really where a lot of the excitement and mystery comes from here, is how they're able to do that. And I wish I had an answer for you on that, but currently we just don't know.
Michael, it's so fascinating to think about this system in its the integrity of it. It's so self-running because once they steal the plasty and they put it in, the kleptoplasty leads to the chloroplasty inside their beings, and they can refrigerate it or whatever the equivalent is to keep it fresh much longer than it would be outside. I think that's just such a fascinating aspect to this that is a mystery. And it leads me to my next question. And thank you for being here and indulging in these questions. When somebody has a curiosity that's as specific as yours, what is the mechanism through which you share your findings on a regular basis? Not big announcements in journals, but is there a network that's free-flowing with information and ideas about discovery at this level? And are there IP concerns along the way because everybody's looking forward to the next grant that they can get and they don't want to cannibalize their own professional trajectory? Can you talk to us a little bit about the culture of research and sharing information, especially information that could be very specific, but if you somebody found out something across the world, wouldn't that ignite you and be exciting? And do you have that kind of flow back and forth?
Thank you, Deborah. It's not exactly refrigeration of the chloroplasts, but that is kind of a fun analogy for that, so keeping them from degrading. Before I go on to answer your question, I did just want to mention real briefly about what I was talking about with the chloroplasts and the mitochondria, that idea about endosymbiosis. That is an ancient event that would have happened long before the evolution of multicellular life. And that's a sort of a theory that explains some of the interesting things that have been observed about chloroplasts and mitochondria. On to your question about how we share information. So the classic sort of model for that for science and is going to be peer-reviewed scientific papers. And that's still ultimately the end goal of any of our scientific studies that we're doing. There's some issues with that model. In terms of accessibility and a few other things, and it's not really sort of the open collaborative sort of thing that you were talking about. Sort of the other end from that is things like public outreach, and I'm a big proponent of science being available to the public. So my lab and I do a lot of things to try to make some of the work we're doing accessible and available. So some of that outreach is fairly simple stuff, like they have an Instagram account, Tampa Slug Life, where I post pictures of interesting slugs and little updates on what we're working on. I do things like interviews like this, podcasts, things like that to talk about our work, and also a fair bit of public talk. So the biggest one of these, that we're one with probably the broadest reach I've done, is the recently published TED Talk on sea slugs, but also do local events. Went to presented at an event called Pints of Science at the New World Brewery here in Tampa, and had a really nice time talking with a lot of people in my community. Had some very neat conversations there. I do talks for scuba diving clubs and sometimes school groups, and probably the most interesting event I've got to take part of in that sort of thing was with an organization called the See and Learn Foundation, which is in the Dutch Caribbean in Saba. I went out there for a week and did some public talks, took people on field trips to look for sea slugs, did some work with school groups and the retirement home there. It was a really, really neat event. Kind of in between those two concepts, the public outreach and the peer-reviewed scientific paper, are things like scientific conferences. So the meetings where scientists are presenting their research to other scientists, and that's really where a lot of that collaboration kind of comes from. At those meetings, you can present work that's not all the way complete, get ideas and feedback from other scientists, and that's really where a lot of collaboration comes from. And so those can be really invigorating events that get you new ideas and new possibilities for directions research could take you in. My field, I would say, is pretty cooperative. So the sacoglossan sea slugs that I work on, it's not a very, very big field. There's not a lot of scientists working on this, and I'd say we all get along pretty well and work together, often collaboratively on projects. But that can be different in other fields of science, particularly if someone is worried about their project being scooped, or if there's a lot of money from a patent or something like that on the line. We don't really have sea slug patents, so that's not really much of a concern, so we can be a more open and collaborative group. But one other thing I did want to mention, since your question kind of leads into this, is the concept of open science, which is the idea of making everything about a research project available, not just the final peer-reviewed paper and a few selected things along the way, like photos on Instagram, but making the whole data set and everything available for other scientists or even the public to look at if they want to do that. And that is something that's gaining some traction. I'm working on a new project, which is not a sea slug one, but rather about seagrass in Tampa Bay. And this one is funded by the Tampa Bay Estuary Program. And one of my requirements for this grant is that everything resulting from it be published in an open science format so that all of this is available for everybody. And I think that's a really good direction for us to push science in so that it is accessible, that the information is there. Other scientists can use it. Other people who...
Hey, Michael, thank you so much for this response. I'm sorry you got caught off a bit there, but I love that grants require that open science mentality when they're giving out their grants, so that people understand that this is a stipulation, and it really contributes to this culture that you're talking about. Speaking of this culture, I want to wrap this up, first of all, by saying I really am happy you're here. And because Swell is set up for images and images and voice and links, it would be great for you to just open up your Swellcast sometimes and just post a picture and say, hey guys, this is what I'm working on. It would be so exciting for us to be a part of your social strategy, because we certainly share information here and want to celebrate the process that you're in of discovery and just understanding what's next for you. So I really appreciate you, and I hope we see you here more because it's kind of exciting. It's such an interesting world that you exist in. It's so unfamiliar to most of us, and what a gift that we're invited into it. Thank you.